ותעשה, אחרי זה תדע לעשות share, ספיקר is not working. חבר לזה, אולי צריך את זה. 1, 2, 1, 2. Okay, so I'll start. So this is a truly fantastic day for me as a kind of scientific father of UDI, and I wanted to kind of say a few words about UDI and you know how we got into the lab and you know the kind of the kind of way or the journey that we went through during this period in the lab. So Udi, I have it. Udi actually met me, or we met. I met him after I gave a talk in the physics department, and he was a second year undergrad student, and he was very interested in the lab and came to talk to me. And then he basically started as a project student in the lab. Okay, have since kind of grown and advanced. Okay, through the years. Okay, becoming later a master's student and becoming a PhD student. Okay, and now even a postdoc for a short period, and I'll tell you about it. Okay. So he's kind of, we went through a very long way, okay, which was very interesting and exciting to go along this journey. And in particular, it was really inspiring to see him kind of grow from an undergrad, okay, all the way to being a full scientist. So just to mention a few words, so he came from physics, and he did his master PhD in biology, biochemistry department, and he basically has managed to do this transition very, very beautifully. And he has been involved in some of the most important works from the lab. He was together with Owen, previous postdoc in the lab, has worked on a paper we had in development of cell that we will talk about. And then beautiful collaboration with Noel Belikuis from Pasteur Institute. Okay, and he basically built really some of the foundations that many of the things that are going on in the lab are actually built on. So it's been a really privilege to have you. And I wanted to say one more thing. So I mentioned Udi has submitted a thesis a month ago, and a few weeks ago. So he's going to be continued for 1 year as a postdoc, but during this year he's going to Paris for 6 months as part of the collaboration we had. And then he will go to a postdoc in Caltech in the group of David Anderson. By the way, I was a postdoc in Caltech, but not in the same group. Different topic. So it's very exciting to kind of see this progression. So I've been. Thank you very much. I want to thank you and tell you that it's been a privilege to work with you and to advise you and just hope and believe that you're wonderful in the future. All right.
Let me just verify.
There's still a problem with the Zoom?
Never mind.
No, but
No,
I don't know.
is that the right Zoom? You make sure that it's the right.
Absolutely right.
Sorry.
Yeah, now it's.
That was the wrong.
Okay. Okay, so hello everyone. Here in via Zoom. Sorry, it was the wrong Zoom link that I opened. Everyone told me to now join. Thank you, David, for the words. Thank you, John. First, just to let you know that I found the recording of the lecture that you gave when I was in the second year, so I can send it to you.
Oh wow!
Yeah. It's on YouTube.
I should be worried to look at what I said about the 6, 7 years ago. Okay.
And so thank you all for joining my PhD concluding seminar. Talk will be about 50 minutes, so let's get started. I will talk about how cell morphology and differentiation dynamics can affect notch-mediated lateral inhibition in the framework.
סליחה, אם אפשר לשפר את הסאונד, זה קצת קשה להבין.
Is it okay now? Okay. So I hope it's okay now. Okay, so let's just get started. Just a short outline. I will give a brief introduction, then I will talk about some of the questions which I will answer with my results, and then summary. Okay. So, general question would be, how do we get from identical cells to differentiated tissues? It could be during development. In this example, this is development of the chick from a single cell to a full organism. And in this example, we can look on the tissue of the chick in the rear. At embryonic day 7, we see a uniform lattice of cells of the chicken in the rear. But if we look on day 10, then all of this tissue is differentiated to hair cells here in white, which are surrounded by spoken cells, which are not marked here in black. And the question is, how do we get? How do we get this differentiation? It could be not only during development, but also in the adult during maintenance of the tissues. In this example, these are the intestinal stem cells, which differentiate and migrate towards the lumen. And in this case, this stem cell pool maintains itself during this differentiation, division and differentiation process. And a key component to these two is notch signaling. Notch signaling is involved in the coordination of differentiation between neighboring cells through a process termed lateral inhibition. It involves many developmental systems from angiogenesis through somatogenesis to neurogenesis and many more developmental systems. So now I want to introduce these two terms, notch signaling and lateral inhibition. Once we understand that, we can continue. Okay. So notch signaling is a signaling pathway that is occurring between two adjacent cells. Okay. If we take two cells from epithelial tissue, for example, we can have one cell that receives the signal. This is the receiver cell that expresses the notch receptor by the notch transmembrane protein. Okay. And we also have the sender cell that expresses the ligand, also a transmembrane protein. And the notch signaling pathway requires the interaction between the ligand and the receptor. So once the ligand binds to the receptor, then the ligand goes through recapitulation from by MID1 of the neuralized, and this recapitulation recruits cluttering. This cluttering induces endocytosis of the ligand. Now the ligand is being endocytosed into the sender cell, and it's still bound to the receptor, so it pulls on the receptor and exposes this cleavage site on the extracellular domain of the receptor. This cleavage site leads to two cleavage, consecutive cleavages of the notch receptor, and it separates it to notch extracellular domain and the notch intracellular domain. Now the notch extracellular domain still bound to the ligand, and it transendocytoses together with the ligand into the sender cell in a process termed transendocytosis. So I will use this term TEC, okay, later on. So they both go through the degradation in the sender cell, and the notch intracellular domain translocates into the nucleus of the receiver cell, where it serves as the transcriptional cofactor. Okay, so this is the notch signaling pathway, but here we only talk about one activation, one-way activation. Only one cell activates its neighbor, but actually we can talk about two-way activation. Okay, each of the cells can express the ligands and receptors, and each of them could be sender or receiver cell. Okay? And this is important for the process of lateral inhibition. In this case, we will talk about the delta ligand. Right? In this process of lateral inhibition, each cell sends notch signaling to its neighbor, but this is an inhibitory signal. Okay? So each cell wants to express the delta ligand and signal an inhibitory signal to its neighbor, suppressing him from expressing the delta ligand, okay, or from activating the delta ligand. Okay. So now we have this competition between the two cells, right? And we can imagine that if we have two cells that are identical and have the same amount of notch receptors and delta ligands, then nothing will happen, right? Because they both will express each other at the same amount and they will stay in this situation, right? But if we have a slight noise slight difference between these two cells, for example, if one of them have slightly more of the delta ligand, then this lateral inhibition process can amplify these differences, and we will end up with two types of cells. Right? We will end up with a high delta cell and a low delta cell, right? Okay. So here in red is the high delta cell, and in green the low delta cell. I will use this color code later on. If we will take this process not only for two adjacent cells, but for a lattice of cells, then each cell in the lattice will try to inhibit its neighbors from becoming the high delta cell. Right? And each cell wants to become the high delta cell. So we will end up in a pattern if there is a slight noise in the initial lattice. We will end up in a pattern of high delta cells that are surrounded with low delta cells. These are the red cells surrounded by the blue cells. Okay. Any questions? Okay, so this is the lateral inhibition mediated by notch signal. If we go back to the chicken here, we can see this in the embryonic tissue, right? We start with the uniform lattice of cells and end up with the differentiated one where the air cells, these are the high delta cells, they are surrounded by supporting cells, these are the low delta cells. Okay. Now, this process of lateral inhibition mediated by notch signaling was first formalized in 1996 by Joan Collier and colleagues. And they modeled this process in a very simplified way, where they only talked about activated delta, which activates the notch receptor. This activation of the notch receptor inactivates delta within the receiver cell, as we saw before. Okay, so they are only just two variables, and then they can express this mechanism in a lattice of cells in terms of differentiation equations, and they can solve it numerically to get the final pattern of salt and pepper like pattern, where these high delta cells in black are surrounded by low delta cells. This was proposed in 1996, and this is a very simplified model. What I wanted to do is take this basic model, or the classical model as I call it, and try to expand it to take into account aspects of the actual biological system that might be important or play an important role in this process of lateral inhibition and pattern. More specifically, I expanded the classical model in three main projects that I did here in the lab. The first project, I took into account the cell morphology. Now, we saw that cells have different size and shapes, and this might affect the notch signaling between the cells and the patterning process. This work was published in 2017 by our group. The second project that I will talk about is a project where I took into account cell dynamics, more specifically division and elimination of cells, and also cell lineage as cell differentiate and change in morphology of the cells. This is a project in collaboration with the group of Laure Bellefeuille from Pasteur Institute in Paris, and we published a paper about it earlier this year. Third project that I will not get into it, but I will just mention it. I'm taking a more complex molecular network of notch signaling that includes not only lateral inhibition, but also cis inhibition and mutual inhibition, and also some gradient of monomer genes in the selection of sensory organ precursor in Drosophila. This collaboration with the group of Thomas Klein from Düsseldorf University. Okay. So let's start with the first project. In the first project, it contains experimental and mathematical parts. The experimental parts were done mainly by Oren Shaya, a former postdoc in the lab, and by Pascal Kamaisi, who is a colleague this year. And some image analysis was done on the chicken in area, as I will show later on. It was done by Dima Rifkin, who is also here, was a former master in the lab. I did mostly the image analysis of some of the data and the mathematical model and computer simulation. If you go back to the chicken here, we can see that cells, I don't know if you can see it, but cells have different size and different shape, and we want to ask how this difference in size and shape and morphology between the cells can affect the notch signaling between them, and how can it affect the lateral inhibition process and the patterning process. Okay. More specifically, we wanted to ask if the contact area between the cells can influence the amount of notch signaling that can be produced between the two cells. Okay. So what we wanted to measure is two things. Okay. We want to measure the contact area between cells, and we want to measure the amount of notch signaling that is being produced between cells. To do this experimentally, we developed an assay where we can track notch signaling. This is the assay of two cell lines. The first cell line, these are cells that express notch secrete, where the secrete flow is on the extracellular domain of the cell. The second cell line, these are cells that express delta and cherry. The angelic fluorophore is fused to the C terminus of the ligand. Now, as I mentioned before, notch signaling occurs between these two cells, and the extracellular domain of the receptor is bound to the ligand and trans-endocytosed into the sender cell. Right? So now these two fluorophores are trans endocytomes together, and we can track it in the microscope, and we can see this puncta of these two fluorophores together, okay, in the sender cell. One more thing that I will mention about this assay. This is a doxycycline invisible assay. The cells that express delta and cherry will start to express the delta and cherry only after we add doxycycline into the system. We can then take movies of this assay in a 3D cold culture, for example. And here we can see this movie where we have the green cells here. These are the cells that express the notch citrine. Right? And once we add the doxycycline, then the middle cells here, it's hard to see, but these middle cells here start to express the delta and cherry. Okay? And as they start to express the delta and cherry, then notch signaling starts to occur between cells, and we start to see these punctas of citrine and cherry coming into the sender cell, into the delta expression. Okay, so this is the transendotitosis disease. Yeah. We also see it here only in the region, in the citrine region. Okay, so what we wanted to do now, so we can measure the amount of signal, right? Doing image analysis, we can measure the amount of signal that's coming into the sender cells, but we want to compare it now to the contact area between the cells in the system. Right? Doing so in a flicker culture will be quite hard to analyze because we want to see if there is any correlation between the two. So what we wanted to do is to develop a system where we only have two cells that are signaling each other. Okay? To this end, we developed a micropattern device. This micropattern device is a device that has these two wells, and we plant cells into these two wells, and sometimes we get a situation where we have one cell that is expressing the citrine, and the other cell will express the delta and cherry, right? And they can contact each other only on one side of this device, and we can measure the contact area between them on that side. Sometimes we get a very small contact area, sometimes we get a very large contact area because the cell kind of undergo the algorithm that owns this device. And now we can measure the contact area between them, and we can also measure the notch signaling that is being produced between them, right? So we have this assay of two cells. We can measure the contact area, and we can measure the amount of trans endocytosis that comes into the sender cell. And we see a correlation between the two, where cells that have a larger contact area, they can produce more notch signaling. Okay? Any questions so far?
היי עמרי קוש, כן, עמרי קוש, היי מתוקיו.
What?
היי מתוקיו. מה שלומך? גם אני אוהב אותך מאוד מאוד ואני...
Yeah, I muted. Okay. So we want to ask how this dependence of notch signaling on the contact area can affect the final pattern or the lateral inhibition process, right? To this point, I developed a mathematical model of lateral inhibition that takes into account the contact area between the cells. Okay, so now instead of looking on cells as individual, what we look in this model, we are looking on the boundaries between the cells. Okay? And we say that if the boundary is small, then some amount of signal can be introduced. If it is larger, then more signal can be introduced in this boundary. And the amount of signal that, total amount of signal that one cell receives is the integration of its boundaries, of signal that it receives from its boundaries. Okay? This model then can be expressed in mathematical equations, and it can be solved numerically. And then we get the final pattern of salt and pepper, where high delta cells are surrounded by low delta cells. So this model alone is sufficient to capture the original or classical model from 1996, right? But it also exhibits this dependency of notch signaling on the contact area. Okay, more specifically, if we look in the simulation, if we look at the time point when we cannot distinguish between the two populations of high and low delta cells, this is a time point that I call tau fade. At this time point, statistically, we don't know which cells are high ligand cells or which are low delta cells, right? But at this time point, we see that smaller cells receive less inhibitory signal. Okay? And this is at the early time point, before the cells differentiate. If at this early time point, smaller cells receive less inhibitory signal, what does it mean about the final pattern? Right? Yes? So I will tell you. So it means that smaller cells have more mobility to become the high delta cells, right? Because they receive less inhibitory signal, right? So they are more likely to become high delta cells.
Yeah. How do you compare the your simulation results to the data that you have?
Yeah.
How do you know that your model has become better at predicting the final pattern compared to the previous one?
Okay, so the question was how can I compare between the model and the actual life system? So I don't compare it directly, but what we did now is to take this model prediction and try to see if it's actually happening in the life system, in the biological system. And if we go back to the chicken here, now the chicken here, these are images that were taken in 1997 from Richardson and Goodyear. Okay, so we contacted them and asked them for images of the chicken here. It was taken in films. They developed it and sent it to us. And then Dina did the image analysis. And what we see is that, oh, sorry, I skipped it. Okay, sorry. Confused me with the, okay, so the model predicts that high delta cells are small. The high delta cells are on average smaller than the low delta cells. This is the prediction for the model, right? And then we went back to the chicken here and wanted to test if this model prediction is happening in the chicken area images. Okay? So we see a bias here where smaller cells are more likely to be selected as high delta cells. And if we go to the chicken area, this is exactly what you see there. Okay, so the pair cells here in white, okay, for many images, not only this is an example. These are the high delta cells, which are smaller, they have a small aperture size relative to the support. Okay. So just to conclude this project, node signaling depends on the contact area between the two cells, and locally, smaller cells are biased to be selected as the high delta. Any questions on this project? Oh yeah, I want to continue to another project. So this was the first project that I published.
Do you want to go today or?
Yeah, we said which
Just let
day.
me know when. Yeah. I'm here.
30 minutes will be okay?
Yeah, perfect.
So you want what?
Just
It's
to.
complicated with you.
Yeah, there's just so much.
To see the results or to discuss or because I have many things to
I can
tell
show
you.
Yeah, I can show you the results. I don't think.
I don't know how to do it, but
I don't know.
that's fine. That's fine. If it happens, I will mute it. Okay. Let's move on to the second project. The second project is the second project is a collaboration with a group of Lobel and Quiff from Storm Institute Paris. The experimental work was done by Nicola Gray and Lomicini from our group. Also, the statistical analysis, as you will see in this project, was done by Felix Jason. I did the mathematical model and the computer simulation. This work was published earlier this year in Cell Sensor. We got a cover, and this cover implies of what we are looking in this project. Okay? We're gonna ask if stem cell differentiation is a coordinated process, okay? Such as these ballerines that are coordinated with each other. This one active ballerine, silence its neighbor ballerines, okay? So we're gonna study if there is any coordination in the process of stem cell differentiation, okay? The paradigm so far is that the process of stem cell differentiation is a completely stochastic process. Some stem cells here marked as A can divide at some rate parameter, and this division can lead to more stem cells that maintain the pool of stem cells, or to progenitors that then differentiate and eliminate from the stem cell pool. But we see evidence that there is some coordination in this kind of process. For example, this paper from 2018 by Mesa et al. They showed that there is a correlation between the elimination of one stem cell and the division of its neighboring cells. Okay, so they see a correlation between the elimination and the division of the immediate neighbors. They suggest a mechanism in which, by the way, this is a mouse skin. These are stem cells on a mouse skin. They suggest a mechanism in which once a stem cell eliminates, because it differentiated from the lattice, then its neighbors take over its space, and there is a growth in the cell size, and this growth might lead to division of that cell. Okay, but it did not show it experimentally, it just suggests this hypothesis. They did show a correlation between it. What we want to ask if there is any combination between the differentiation of stem cells, and specifically we are looking on neural stem cells in the adult zebrafish brain. Okay? In the adult zebrafish brain, stem cells are glial cells that form a monolayer of cells around the brain, okay, on the outer layer of the brain. Okay? And these glial cells, the neural stem cells, they can divide and they can generate progenitors, which then differentiate into neurons and descend into the brain. Okay, so we have this monolayer of glial neural stem cells that divide and differentiate, and the neurons delaminate from this layer into the brain. Okay? Okay. What we wanted to ask is what is the mechanism that keeps this pool of stem cells in homeostasis? Because some of the cells divide in order to generate more stem cells. Okay, and some of them will differentiate into the neurons. I want to see if there is any mechanism that is coordinated. Okay? We will use these four approaches. We will use static imaging, live imaging, where we take a live fish and image the brain once every few days, statistical analysis, and mathematical modeling and computer simulation. So we first wanted to see if there are any or distinguish between different states of the neural stem cells along the cell lineage and they differentiate. Okay, to do so, we use static imaging. We did, Nicola did all-mount immune staining for adult transgenic fish. Okay, they used a GFP marker for glial cells. Okay, this is the GFAP here in green, so they can mark glial cells. So I mentioned that the neural stem cells are glial cells, and they can see it here, these are the green cells. Okay? They also use a marker for cell proliferation. This is the purple marker here, PCMA. So they know which cells can now proliferate. Okay? So we have these two kinds of markers. They take an image of all of the population of the neural stem cells in the brain, and they now can distinguish between three populations, okay, three types of cells. Okay. The first state, these are what I call the QNCs, quiescent neural stem cells. These stem cells express only the GFAP marker, but they cannot proliferate at the moment, so they do not show the PCMA marker. Okay, so these are quiescent neural stem cells. The second state is the activated neural stem cell, which are stem cells that they are still glial cell, right? They still have the green marker. And they also have the PCMA marker, which means that they are now active and can divide. Okay. And the third kind of cells, these are AMP cells, activated neural progenitors, they are no longer linear cells. They do not show the GFP marker. Okay, so they are no longer neural stem cells, but they are progenitors that can still proliferate. So they only express the PCNA marker. Okay, and this cell will eventually differentiate into neurons. Yeah. So these cells are related to each other. Okay, so we have these three cell states: the QNSCs, the ANSCs, and the ANPs. I will use this color code, this green, purple, and orange during the rest of the talk. And these states are related to each other by a lineage. QNSC can become active. This activated neural stem cell can divide to generate more neural stem cells or progenitors that will eventually differentiate into new ones. We also have a direct path where QNSC can directly differentiate into neuron, yet this is a rare pathway compared to the main cell lineage that includes the division. Okay? And we want to ask if there is any correlation between the different cell states. Okay, are there any correlations between the different cell states? More specifically, we wanted to ask if there is any spatial correlation between the positioning of the different cell states. To answer this question, we took the images of the neural stem cells and we also extracted the positions of the neural stem cells in space. Okay, so we did the image analysis. And we have the center position of each of the cells. So now we have two kinds of information. We have the position of the cells and we have the state of the cells. Right? And we can now ask if there are any, we can compare the distribution of the different cell states in space. Right? Okay, the way we do it, I will just draw an illustration, is by asking how the position of one population affect the positioning of another population. So in this example, we ask how the positioning of ANSCs affect the positioning of the same population of ANSCs. Maybe ANSCs affect the positioning of other ANSCs, maybe not, okay, maybe just random. Okay? To try and answer this question, we took the image and we used the statistical function that is called L function or Besak's L function. I won't get into it too much, I will just explain what we see here. So this L function is a function of the distance from the cells that we are interested in, right? And now we want to compare the positions of ANSCs compared to the position of other ANSCs, okay? And I want to say if it is just randomly scattered around, or maybe they are clustered together or more dispersed. Okay, to answer this, we use this L function, and now we need to determine what is random. Okay, to determine what is random, what we did is taking the positioning okay, from the image, and just I don't see anyone talking actually. Okay, it's over now. Okay, so what they did is taking the positions of the population, the ANCs, and they just scatter it randomly around the positions of other cells in the lattice. So this will be one random perturbation, where you just randomly scatter the cells around the lattice. Okay, so they did 1,000 of these perturbations, and they measured or they calculated the L function for each of these perturbations, and then we can create this randomness tube that contains 95% of the L functions from the random perturbations, right? So then we can measure the L function of the actual image, right? And this would be this red curve. If this red curve is inside the randomness tube, it means that 95% that it is a random perturbation, right? Just a random scattering of the cells. Okay, so what we see here in the case of ANCs compared to ANCs, we see that it is random, okay, which means that ANCs do not influence the distribution of other ANCs. Okay. Okay, so now we check the same thing between ANCs and ANPs, okay, progenitors compared to activated neural stem cells. And what we saw is that there is a dispersion at about 1 to 2 cell diameters, which means that the ANPs and the ANCs statistically do not neighbor each other. Okay? So there is something that keeps ANCs away from ANPs. Okay, and what is it? What is it? Okay. Okay, so this is spatial correlation. And what keeps ANCs away from ANPs? Now I remind you that my talk is about notch signaling and not transmission. And we see this behavior where one cell keeps its neighbors from becoming some cell states. So maybe it got to do with signaling. Okay? How can we measure it? We can also track the delta ligands in the system. Okay, and what we see is that QNSs in the system, okay, do not express the delta ligand. Yet ANSs do express the delta ligand, and more importantly, ANPs express the delta ligand. Okay, so it might be the case that ANPs inhibit their neighboring QNSs from becoming activated neural cells. How can we test it experimentally? One way to do it is by blocking notch signaling altogether, right? If we block notch signaling, then we can expect that if it is true, then the dispersion, this correlation will be lost, right? Okay. So what will happen if we block notch signaling? We do the same analysis in the wild type, we see this dispersion, right? Once we block notch signaling for 24 hours, then this dispersion is lost. We lost this correlation, and it is just a random distribution of ANPs compared to ANCs. If we look at the control, then this dispersion is back, this correlation is back. Okay. So I conclude that ANP might inhibit their close neighboring QNSs from becoming activated ANCs, activated neural cells. Okay. So this is spatial correlation between the two, and now we wanted to ask: is there any temporal correlation as well? Not only spatial correlation, but also a temporal correlation. To this, we used live imaging, okay, of the brain. So Nicola took the fish once every 3 days and imaged the whole brain, and you see here from the imaging the skin, the skull, and also the neural stem cells here in green. Okay, and then we can we can start and track the neural stem cells, right, over time. Yeah, we can track their state. So we have this image. In this imaging method, they can only distinguish between quiescent neural stem cells and activated neural stem cells. So they cannot track the progenitors, but it's enough for our case. And they can generate these movies. And in these movies, we can start to see events. For example, this division event, where one cell divides into 2 cells. So from day 6 to day 9, this cell divides because we see a pair of cells instead of one after 3 days, right? And we can also see differentiation where one cell just delaminates and disappears from the lattice. Okay, so we see these differentiation events. Okay, so now we can see this division and differentiation events, and this is from only one cell, but we can take it from many cells and do the segmentation, and we can generate these tracks for each cell, and each cell we have the time axis, and we can see the state of the cell which the cell is in, right? And we can also see these division events. Okay, so we see these division events. This, for example, the most far away track. Okay, and we also see differentiation events. Okay, where the cell disappear from the lattice. Yeah, so we can track these cells and the state and division and differentiation events, and specifically we are interested in the division events. Okay, so we are looking on the cells that are about to divide. We know they will divide in the next 3 days because we see pairs of cells in the next time step. We call this cell that is about to divide a mother cell. Okay, because it is about to divide. It's not an actual state, but it's actually an activated neural stem cell that is about to divide. Okay, and we want to see if there is any correlation between division events, okay, or between mother cells. But not only at the same time, we want to see if there is any correlation also in different times, okay? So specifically what we did is looking on mother cells at time t, and we asked the question, is there any correlation between division events between mother cells at time t with other mother cells? It will be at the same time, but not only at the same time, maybe it has correlation with mother cells at t plus 3 days, or t plus 6 days, or t plus 9 days, 9 days so far, right? So this t plus 3 days, we call it delta t1, okay? The question is there any correlation between the positioning of mother cells at time t with the positioning of mother cells at time t plus delta t1, okay? Or delta t2, or delta t3, and so forth. And we use the same statistical function, the L function, to measure these correlations if there are any. What we see is that it is random in all cases, okay? Which means that there is, apparently, it means that there is no correlation between the positioning of mother cells at time t with the positioning of mother cells at any other time. But this is only for one fish, and in this fish, also in all the other fish, what we see is at delta t4, there is a drop, a significant drop in the p-value, okay, in the chance that it is random, right? So it might be a weak effect here, okay, which is not significant for one fish. So what we did is doing the statistics on all of the 3 fish. And if we do so, we see that there is a significant correlation at delta t4, which means that mother cells at time t, the positioning of division events at time t, influence the positioning of division events around them, but not at the same time, yet with a time delay of 9 to 12 days. So mother cells at time t affect division events only 9 days afterwards. Okay? And also at the range of 1 to 2 cell diameters. Okay. So we wanted to ask then, is this spatial temporal correlation is also notch mediated? Experimentally, we cannot answer it, because if we were to block notch signaling for 9 days, the fish will die. Okay? So our approach will be using a mathematical model that takes into account the cell lineage of the neural stem cells. So this is the same cell lineage as before. These QNCs become active. This activated neural stem cell can then divide through the mother cell state, right? Quasi-state, yeah. And it can divide into more QNCs or to generate progenitors. These progenitors can divide once again or differentiate into neurons, right? We also have the direct path where a QNC can differentiate directly into neurons, and then we can assign parameters for each of these transitions, okay? And we have these gamma parameters, which are the rates of each transition, right? It is the rate for QNC to become active, the rate for the dividing. We also have the probabilities for dividing into different division states, right? And one thing I will tell you about it is that using the mathematical model, expressing it using mathematical equations and using the sum of the data, we were able to extract all of the parameters in the system, which is quite rare thing to do in general. So we have all of the parameters, the actual parameters in the biological system. Okay? Then we can simulate this model using computer simulation, and we can simulate these different cell states. Okay, these rings are QNCs, ANCs in purple, ANPs in orange. Okay, and we can see these different events that occurring during the lineage. For example, from T1 to T2, this QNC cell become active. Okay, and from T3 to T4, this activated neural stem cell divides into one progenitor and one QNC cell. And from T4 to T5, this progenitor cell differentiates and eliminates from the lattice. Okay, so we also include in this model mechanical changes, okay, or morphological changes where cells can divide, they can eliminate, okay, they can change their size. And now I will show you a movie that capture it. Okay? So this movie is for two simulations. One simulation is a simulation with lateral inhibition. Okay, I forgot to mention it, but we have this lateral inhibition here between ANPs and QNCs. The ANPs prevents their neighboring QNCs from becoming active, right? And a model without lateral inhibition. And I remind you that we want to ask if this lateral inhibition can generate the spatial temporal correlation. So we want to compare these two models. So I will play the movie just to show you that this simulation can capture the different events. Okay, so it will run quite fast. Okay, but it's just to show you this. Okay, so you see that cells can change state, they can divide, they can eliminate. Okay, they can change their morphology, their size. Okay, and we can do it for a model with lateral inhibition and for a model without lateral inhibition. Okay. All right, and then we can plot the analysis of these movies, of this kind of movies. Okay. For example, we can take 18 such movies and do the analysis from day 300 to 500. I remind you that the parameters are in the units of the actual biological system. Okay, and we simulate for 200 days, for example, 500 days. I already show you this last 200. And what we see that the model remains at steady state or in the number of cells, okay, and also in the ratio between the different cell states. Okay, so there is a steady state that the model approaches to and stay like this for many time steps. And the model without lateral inhibition also exhibits this kind of steady state. We adjusted the parameters such that the model without lateral inhibition also become steady, okay? Because we want to compare between these two models, we want to compare the spatial distribution between the two models, so we had to do this adjustment of the parameters. These two models capture the exact cell states as observed experimentally, so we have the same ratio between the cell states, okay, in this both model with lateral inhibition and without lateral inhibition compared to the right. Okay? And not only the same cell states ratio is captured by the model, but also the different division events or differentiation events. Okay, the ratio between symmetric or asymmetric divisions and the differentiation is captured by the model and it fits the biological system. Right, so the model captures the dynamics of the biological system. That's what I want to say. Yes. And the model also captures the lateral inhibition effect from ANPs to ANSs. No surprise because I introduced it in the model, right? So a model with lateral inhibition generates the correlation that we saw before, where ANPs keeps their ANCs away, right? And without lateral inhibition, this correlation is lost. So now is the question, can this model alone can capture the spatio-temporal correlations where these model cells or division events keeps division events from happening around them with a time delay of 9 days. And what we see is that the model capture the exact effect with the exact time delay, okay, where mother cells at time t suppress other division, other mother cells to occur at time t plus delta t4. Right? Okay, so the model alone is sufficient to capture this behavior. What happens if we were to remove lateral inhibition? Then this dispersion is lost at delta t4. Okay? So I conclude that the spatial temporal correlation is not dependent from the model. Okay, and now we can ask, okay, but why is there a time delay? Because node signaling occurs at the same time. There is no delay there. So where this delay comes from? So I remind you that this is a cell lineage, okay? And some cells stay at a certain state for a certain time. And more specifically, ANPs are the one that lateral inhibit their neighbors. So maybe the amount of time that ANPs are in the system generates this kind of a delay because they are then eliminated from the lattice, okay? So what we did is to test it. So we increased in the model, we increased the rate of which the progenitors differentiate from the lattice or divide, okay? If this is true, and this rate of the A and P's differentiation influence the time delay, then I expect that the time delay will get shorter, okay? So higher A and P's dynamic rates will generate a shorter time delay. This is my expectation. Specifically, I multiply these rates by 4/3, okay? Which means that if by chance this there is a linear relation between the rates of differentiation of A and P's and the time delay, then by multiplying the rates by 4/3, the time delay should be shortened by 3/4, okay? So the time delay should move from delta T4 to delta T3, and this is exactly what we see. The time delay just shifts from delta T4 to delta T3. Okay. So just to conclude this, neural stem cell differentiation is coordinated by the coupling of lateral inhibition and the cell lineage itself. Okay, we need these two to generate the exact time delayed correlations. Just last thing I want to say about the significance of this kind of mechanism. We can generate simulations where we plot the positioning of neurons, of the differentiated neurons over time. So at each time step, we just plot the position of the differentiated neurons, and then we have these layers of neurons over time, right? So now we have a 3D plot of the differentiated neurons. So we have the distribution of the neurons in the brain in the 3D, right? In some layer of the brain, okay? And we wanted to ask what is the importance of having this lateral inhibition and the delayed correlations. And the importance is that a model with lateral inhibition compared to a model without lateral inhibition generates a more sparse or less clustered pattern of neurons. So it is important to make the generation of neurons across the brain more uniform than without lateral inhibition. Okay, just to conclude this project, neural stem cells differentiation is coordinated through node signaling from the progenitor cells on the quiescent cells. And there is a coordination that leads to spatial temporal correlation that is originating from the coupling of the lateral inhibition with the cell lineage itself. Okay. Long story, right? But do you have any questions about it? That's good. Okay. So you can take questions, of course, afterwards. So this was the second project that I included cell dynamics in the cell dynamics with lateral inhibition with the same system. And the future work of mine will be combining these two projects together. This is what I will do now, as David mentioned in Pasteur Institute. And a third project, which I will not get into it, so let me just press the skip button. So to summarize the whole thing, I started from the classical model of lateral inhibition from 1996. I expanded it in 3 ways. Okay, one project took into account the cell morphology, and we saw that it creates this bias in the selection process where smaller cells are biased. This was published. Smaller cells are biased to be selected, right? This was published as I showed you. Sorry. And the second one was integrating lateral inhibition with cell lineage during homeostasis or maintenance of stem cells pool in the adult system. Okay. And this was published earlier, as I showed. The third project I did not talk about, I will be happy to present you this on another occasion. Future plans will be published in the third project that we're still working on. And this is a collaboration with Tom Klein from Düsseldorf University, and to continue a collaboration with Laure Bellec, combining these two models from the first one with the contact area and this one with the cell lineage. So this I will do in the next few months. So just wanted to thank everyone. So thank you all of the Springs of Lab members from the present and past. We did a long run with many people coming and going. I want to thank my PhD committee, Dr. Friedman, Professor Glodar, and Professor Belson.
Professor Dino Friedman.
Yeah, oh, sorry.
She just received it.
Congratulations. And of course, I want to thank you, David, for everything. It's really it's hard to include this all long run that we went through, but I wanted to tell you that I learned a lot during this day. I learned a lot from you, and I hope I really felt it. Really thank you for everything you did for me and support for now. So I want to thank you about it. Also thank my family, my parents, and my wife, and also my brothers who are not in this picture. And first of all, I want to thank you for coming and joining the seminar. And feel free to take any questions. If you want. Also at home, if you like, you can now unmute. It's good, it's good.
מזל טוב.
אודי היה ממש טוב.
טוב, טוב.
היה הרצאה ממש טובה. אחלה, דיברת את זה מאוד יפה.
אני ראיתי בזום כל הדרך. ראיתי שהיו 3 חלקים, אז אמרתי לפחות השלישי אני אספיק, ואז לשלישי אתה עושה לי סקיפ. אני באמת מצטער, הייתי הבוקר.
לא משהו שלא ראיתי.
כן, ובכל זאת רציתי להיות פה. היה נהדר.
זה מצחיק, 7 שנים אני איתה מחלת ברור, כן. אני כבר קניתי, אני מביאה.
איפה אפילו פודיום?
כן, כמה אם הייתי רוצה.
תקשיב, זה היה מאוד ארוך, החלק הראשון החלק השני היה קצת קשור.
מה ששאלתי, שנייה יכולתי להציג
לא, זה חשוב.
טוב, אז אתה ממשיך ללמד אותם קורסים?
לא, אני לאט לאט שם מתקדם, אני עכשיו קיבל טיפוס משלי של איגוד.
סבבה.
סוג של פשוט. סוג של אותה דרך, אבל...
יפה.
האמת שאני מחפש פרויקטים לסטודנטים שלי בשנו, מעניינים לעשות את פעולה, אולי מחשבות כאלה של גודות, כן, שהם יעשו איזשהו פרויקט במעבדה, כאילו משהו שיהיה קטן, אם יהיה לך רעיונות.
כן, לגמרי.
לי ונבדוק. מדי פעם עושים פרויקטים כאלה, אבל עכשיו פשוט יש לי די הרבה פרויקטים.
אני כאילו חושב על זה גם בדיעבד, הטעות הכי גדולה שעשינו עם העיבוד תמונות שאני עשיתי.